for accepting to give us to share his work and to give this talk. It is a pleasure to introduce him. He graduated from biology at the Federal University of Rio, and he continued on the graduate school doing his master's and his doctoral training with Dr. Sergio Teixeira Ferreira. He did his postdoc here as well at UFRJ, and during both his training as a doctor, as a PhD student and a postdoc, he went a few times to the US, to New York, to continue some of his work at Columbia University, usually working on the aspects of neuronal stress in Alzheimer's disease. He started as a professor in 2018 as an assistant professor at the Medical Biochemistry Department Institute here at UFRJ. And along his career, he has received several prizes and awards. I'm going to mention just a few because if not, then we're going to stay here talking more about his awards than his talk himself. So he is a productivity fellow at the CNPq level 2. He's a young scientist of our state through FAPERJ. He has been awarded a Serra Pilheira grant in 2021. He has also received the Blas Frengen Early Career Achievement Award in 2022, as well as nominated as the 11 early career researchers to be watched by Nature Medicine. He has been affiliated to the Brazilian Academy of Science in 2021. He's a member until now, and he has been very productive, even though he has a young career. He has a factor age of 18 with more than 30 research articles published. He has written 2 chapters from books, and he has worked a lot in the scientific talking about science also within the general media, not only scientific media. Has worked a lot on human resources as well, training both undergraduate and graduate students. Now he has 5 undergraduate, 2 masters and 2 PhD students, as well as 2 postdocs in his lab. And he has already helped graduate 10 other students. And so just to make it brief, and now I can give the floor to Michelle. So thank you again, Michelle, for accepting our invitation and to bring the metabolic inflammatory alterations in Alzheimer's disease to our group here.
So thank you very much, Miriam, for the kind invitation and for the kind introduction. I think it's much more than I deserve and I did, actually. But this is a different audience, so I'll try to keep it short, and I'll try to show a bit of what we have done in terms of interface between inflammation and metabolism and Alzheimer's disease. I know that some of you may find it different to see me here presenting about Alzheimer's disease, because that's probably not what most of you work on, but I'm happy to take any questions during and after the presentation. And of course, immunity and inflammation have been at the center of many disorders. So I think it's important for us to look at the whole potential of inflammation. Okay, so my lab has tried to, since my training as a graduate student and a postdoctoral fellow, I try to understand what happens in the brain that causes memory loss in Alzheimer's disease. And I will start with let me use the laser pointer here. Okay, I'll start with a brief introduction. Then I will show a couple of results showing what we demonstrated in the past relating to how an inflammatory pathway causes memory loss in Alzheimer's disease. Then I'll move to more recent work showing that there is potential for neuroprotection if we treat either the inflammation or stimulate hormonal signaling in Alzheimer's disease. And finally, I'll talk a bit about the recent work about lipoxy and aging and Alzheimer's disease that had the collaboration of Claudio Canetti from Instituto de Biofísica. And with that, I think I can close my presentation along these three major works. Okay, so as almost everyone knows, that memory loss is the main symptom of Alzheimer's disease. But what most of us don't know is that it is essentially idiopathic, which means that there's no known cause or specific cause for memory loss in Alzheimer's disease. What we know so far is that age is the main risk factor, and unfortunately, there's no effective way of diagnosing it or treating it. Of course, over the past 30 years, we advanced a lot in the field, so we now know much more than we thought, than we knew before. But we still don't have a cure or an effective diagnosis for Alzheimer's disease, and that's the question that we're trying to address in our lab. And of course, there are many other labs worldwide trying to look at it. Okay, dementia and Alzheimer's disease are a health problem worldwide because its numbers tend to increase over time. So this is a projection. There's almost 15 years since it was established, and it's been closely followed. So it seems to be right. In, let's say, 30 years, we'll double or triple the number of Alzheimer's disease cases worldwide. And of course, there's a cost associated with it. There's a financial cost, but there's a social, emotional, and affective cost associated with the higher burden of Alzheimer's disease. And it's also important for us here in Brazil because the prevalence tends to increase, but it tends to increase more in low and middle income countries, which is the case of Brazil. So we have to do something here. We have to try to find preventative strategies, and we have to try to find effective therapeutics for dementia. So I think that's a major reason for me to study Alzheimer's disease. I recently had an aunt that died of dementia, but I have studied Alzheimer's disease since I was an undergrad, and I think it's important for us to do something for our country and, of course, for the world. So if I only had 5 minutes or less to talk about what happens in Alzheimer's disease and what we know of Alzheimer's disease today, I would show you this slide. So that's the sequence of events that develop in the Alzheimer's disease brains. And as you can see here, there's many things happening before clinical function and memory is impaired. So when memory starts to become impaired and clinical function starts to decline, many molecular events have happened in the brain that cause neurotoxicity, and that's what we have to target. So we have to understand what happens at the beginning of the disease before memory or any clinical symptoms occur to try to have better options of interventions. And that's something that I generally say that looks like cancer. If you go and treat a cancer patient after metastasis, it becomes much more difficult than if you early detect it and treat it early. So that's what we're trying to do here. And I want to bring your attention to this red curve here, showing that this A beta, which is a peptide that accumulates in the brains of Alzheimer's patients, it's the first known or hallmark of Alzheimer's disease. Then there's tau accumulation and tau-mediated neuronal injury. There's problems with brain structure. So it means that patients start to lose brain weight and brain volume. And then the memory and clinical function symptoms begin to develop. Okay, so I just told you that the first event in Alzheimer's disease is the accumulation of this A beta peptide. So this A beta peptide is an amyloid peptide that is derived from a precursor protein called APP, amyloid precursor protein. It tends to aggregate a lot, especially the version with 42 amino acids. It forms soluble small aggregates, we call them oligomers, and it also forms fibrous, large fibrous. And these fibrous deposit into amyloid plaques, as you can see here. So when the German neuropathologist Alois Alzheimer first described Alzheimer's disease, he described it as a disease of memory with plaques and tangles. So he had access to some let's say, neuropathological or pathological techniques at the moment. And he described these lesions called senile plaques at the time, and now we call them amyloid plaques. But in addition to these fibrils and these lesions that deposit in the AD brain, we have these soluble oligomers that are able to diffuse through the brain parenchyma. And it is able to bind specifically to synapses and to trigger synapse impairment. So these amyloid oligomers, they have many receptors here at the synapse, at point of contact between two neurons, and they disturb the signaling pathways that normally would occur here, and they impair the communication between two synapses, the two neurons. So that's our main hypothesis or our main approach to treat Alzheimer's disease now. We have to block the amyloid interaction here at the synapse and have to permit correct neuronal function. So when these A-beta oligomers bind here to the synapses, they trigger oxidative stress, they trigger tau phosphorylation, and of course they trigger synapse failure and loss. And that's what we thought that underlie memory problems in Alzheimer's disease. Okay, so that's a hypothesis. There's many experimental evidence supporting it, but of course there are others. But if one was to ask me what causes Alzheimer's disease, is it a genetic disorder or is it a sporadic disorder? Well, most of the cases are sporadic. Of course, there are genetic variants that increase or decrease the risk of developing Alzheimer's disease, but the hereditary components of Alzheimer's disease is not that huge. So we have some mutations in APP and in other enzymes that process the APP protein, but most of the cases or most of the risk is caused by some mutations in other genes that increase the risk of sporadic Alzheimer's disease. So we now have the TREM2 mutation that has received much attention recently. We have the ApoE4, which was described almost 30 years ago and still receives considerable attention to. And of course, we have many more less frequent mutations that also contribute to the risk of Alzheimer's disease. But all of them, including this hereditary component of Alzheimer's disease, all of them have in common the fact that they somehow are linked to inflammatory pathways. So they somehow affect microglia in the brain, and somehow they trigger release of inflammatory cytokines that contribute to pathogenesis of Alzheimer's disease. So in terms of epidemiology, what are the risk factors for Alzheimer's disease? So we have many prescribed risk factors for Alzheimer's disease, but I want to bring your attention to this red or pinkish square here, showing that inflammatory mechanisms or inflammatory disorders such as insulin resistance, type 2 diabetes, obesity, heart disorders, they all increase the risk of developing Alzheimer's disease. So that brings us to a scenario in which mechanisms or conditions or diseases that promote inflammation, they increase the risk of having Alzheimer's disease at the end of our lives. And in terms of mechanisms, my past supervisor, Fernando de Felice, and others, many others, showed many years ago that there is a brain insulin resistance phenomenon in the Alzheimer's disease brain. So in terms of molecular signaling, it's quite similar. It resembles a lot what happens in diabetes. So there's increased circulation of cytokines, activation of stress kinases, phosphorylation of the insulin receptor substrate, IRS, in different residues that block its function. So it happens in the brains too, but instead of controlling glucose metabolism and glucose uptake, it controls synapse function. So it's another way of thinking how defective insulin signaling may trigger deficits in the brain rather than peripheral glucose impairments. And there's a vast literature in that. There are many labs that showed similar evidence over the past 20 years. And of course, there is metabolic defect in Alzheimer's disease too. If you look at the PET scan of a control patient and an Alzheimer's disease patient, there is selective hypometabolism. There's selective reduction in the uptake of glucose in regions in the brain that are important for memory function. And this parallels the accumulation of beta amyloid. So that's a probe for beta amyloid, Pittsburgh compound, that increases in the Alzheimer's disease brain, and while actually there's reduction in glucose uptake in the brains of Alzheimer's disease patients. So we have this scenario in which there is beta amyloid accumulation of a beta peptide, there is metabolic disturbances, and there is inflammatory responses in the brain. So almost 10 years ago, we demonstrated that TNF-alpha, the cytokine TNF-alpha, is important to cause neuronal impairment and memory impairment in Alzheimer's disease. So this is an experimental paper that was published during my PhD, and we showed that these A-beta oligomers, which we term A-beta O's, they increase the activation of a stress kinase called PKR, and this is blocked by infliximab, which is an antibody that blocks TNF-alpha signaling. And in collaboration with Lício Veloso at Unicamp, we showed that when we deleted, or mice that had the TNF receptor 1 deleted, they were not impacted by the effects of A-beta oligomers. This is a memory test that I don't have time to show you, to explain to you, basically. But basically, there's this distinction between a novel and an old object, something that the mice knew before, and some object that the mice didn't have any previous contact with. So if it discriminates, if it forms memory, a mouse will discriminate between these two objects. So as you can see from vehicle wild-type treated, wild-type mice here. So they discriminate between the old and the new object. When we inject A-beta oligomers into the brains of wild-type mice, they can no longer discriminate between old and new objects. But if they are knocked out for the TNFR1, they can again discriminate. So that means that TNF receptor and possibly TNF-alpha signaling mediates memory impairment triggered by A-beta oligomers in the brain. And that's true for another contextual fear conditioning test. So that's what we showed, that's part of what we showed almost 10 years ago, and that implicated TNF-alpha signaling in the memory problems, the memory impairment caused by A-beta in the brain. And so this is the whole signaling pathway that we dissected at the moment. So we show that A-beta oligomers promote TNF-alpha signaling, binding to TNF-alpha receptor 1. It activates PKR, which is a stress kinase, and PKR has a double effect here. On the one hand, it triggers insulin resistance through IRS1 serine phosphorylation. And on the other hand, it triggers phosphorylation of a translation factor that blocks, reduces, attenuates protein synthesis in the neurons, and it in turn causes synapse loss and memory failure. And that was the cover of cell metabolism almost 10 years ago. And in the same paper, we show that using anti-diabetic strategies such as GLP-1 analogs, we could protect mice from memory impairment caused by Alzheimer's disease. So these mice are transgenic mice that harbor a mutation in APP, a human mutation that causes Alzheimer's disease in humans. And these genetically engineered mice, they develop memory impairment that is age-dependent. But if we treat them with anti-diabetic agents, such as exendin-4 or liraglutide, which are GLP-1 analogs, they are protected from memory impairment initiated by this mutation. So that tells us that there's a window, or there's a potential opportunity to use compounds that have been already approved for human use, such as these anti-diabetic agents, to treat memory problems in Alzheimer's disease. Of course, this is a mouse study. We also did some studies in non-human primates too, but the relevance of this hypothesis and the relevance of these potential therapy for humans still has to be understood. And what made us happy was to see that this paper and other papers in combination, they encouraged some companies to do some clinical trials in humans. And there are clinical trials now in the UK and in Denmark to test this possibility that liraglutide and examine for could be important for human therapy. And that's really important because although we had recent approvals of compounds to treat Alzheimer's disease, none of them is the cure or is the best effective, the most effective treatment that we wanted to have. So we're still in search of a potential treatment. And in addition to our interest in pharmacology or in pharmacological approaches to treat Alzheimer's disease, we also looked at non-pharmacological approaches that could be interesting. And of course, what popped up into our minds was exercise, because physical exercise is important for everyone. It prevents a lot of disorders. It prevents heart disease, cancers, some types of cancers, metabolic disorders, and it's also important for the brain. So in persons that have no, let's say, comorbid or any health conditions, exercise improves memory, so physiological memory. It increases attention, it increases neurogenesis, it promotes the release of several molecules that are important for brain function. So it's all benefit in the end. And it has not many recommendations against exercise. So that's something that everyone should practice. We often practice less than we should, and we would benefit much more from exercise if we practice more. So we wanted to understand what is the potential of physical exercise to trigger neuroprotection in the brain, especially in the context of Alzheimer's disease. So there is some clinical evidence showing that walking or regular exercise stabilizes cognitive function in patients. So it's not a cure. It's not the final way of halting or even reversing Alzheimer's disease, but has some protective effects. And of course, people that practice regular exercise across life, they are, let's say, protected from Alzheimer's disease. They have a reduced risk of developing dementia. So there's something in there. So we wanted to understand the neuroprotective effects of exercise in the brain. And we again turned into mouse models to try to understand what happened in the brain upon exercise. And we focused on this myokine called irisin, which was described 10 years ago or so by Bruce Spiegelman, Harvard University. And this myokine was initially reported as a myokine, as something produced by the muscle, released into circulation, and that would reprogram the adipocytes or the adipose tissue to favor metabolism. So irisin is produced from a precursor protein called FNDC5. It is cleaved. This precursor protein is a membrane protein that is cleaved into irisin. Irisin is released into the circulation, and it has the potential to affect many tissues. That's what we now know. And so exercise increases the blood or plasma content of irisin. And when mice that are obese are administered irisin, they have a slight but significant reduction in glucose intolerance. So it has some important metabolic effects. And a follow-up paper by Spiegelman lab also showed that irisin, or the precursor protein, actually is expressed in the brain, the hippocampus specifically. The hippocampus is a brain region that's very important for memory formation. So it means that FNC5 is expressed in the hippocampus, and it is also increased in the hippocampus upon exercise, as well as in muscle, as we would expect. So at the time, there was basically that was the evidence in the literature of a potential role of irisin in the brain. So we turned out to study what happens in the brain, especially in Alzheimer's disease, in terms of irisin signaling, and of course, in terms of the potential of irisin to trigger neuroprotection. Okay. So in collaboration with Joe Absambra at the University of Kentucky at the time, we showed that there is a reduction in the content of irisin in the brains of Alzheimer's disease patients, but specifically in the brains of those at a later stage of disease. So early AD patients with slight memory problems, several memory problems, had no difference in terms of irisin. But later, these patients at an advanced stage of Alzheimer's disease had a reduction in the hippocampal contents of irisin. And in collaboration with the DOR Institute here in Rio, with Fernando Mall, we showed that AD patients had reduced levels of soluble irisin circulating in the cerebrospinal fluid. So, and this was also true for patients with dementia with Lewy bodies, which is another form of dementia, a bit different from Alzheimer's disease, but also an important form of dementia. So it looks like patients with dementia, they had reduced central irisin, so reduced irisin in the brain. So that led us to investigate whether reversing this phenotype would be important to protect mice from memory impairments. But before we did that, we also found that the content of irisin in the cerebrospinal fluid correlated with memory. So this MMESE is a memory test used in humans. And we show that the lower the score in this memory test, the lower the content of irisin. And the lower the content of CSF A-beta-42, that A-beta peptide, that amyloid peptide, the lower the irisin levels. You may be asking, well, so if there is reduced A-beta-42, there is more protection against Alzheimer's disease, right? No, that's false, because the levels of A-beta-42 in Alzheimer's disease patients, they build up in the brain, but they are reduced in the CSF, they are reduced in the cerebrospinal fluid. So it aggregates in the brain, it is not dragged out of the brain, and the levels in the CSF remain low. So it means it's a negative correlation with the memory phenotype in Alzheimer's disease. So it looked like somehow, of course, these are all correlations, it's not causal, but it tells us of a potential importance of irisin in terms of neuropathology, as observed by beta amyloid content, and of course, memory, as noted by this memory score. Okay, so we have this scenario in which patients with memory impairment, patients with dementia, they have reduced levels of irisin in the brain. So what if we replenish, what if we recover the levels of irisin in the brain? Would we get a preserved phenotypes? Of course, we have to start with mouse studies. We cannot do that in humans at first. And that's what we did. We started with mouse models by overexpressing, using an adenoviral vector to overexpress this FNDC5, which is the precursor protein of irisin in the brains of Alzheimer's mice. And we got a good level of expression here. We almost had 60 to 7% of increase in the content of FNDC5 mRNA in the cortex. And it was really effective in preventing memory impairment in Alzheimer's mice. So mice, wild-type mice that had A-beta oligomers, and they had a control vector, they could not distinguish the old and new object in that memory object recognition test. But mice that had A-beta oligomers and the expression of the FNDC5 were fully protected and could distinguish normally between the old and the new object. And that's the same in another task here, which is the contextual fear condition. So it was our first among many lines of evidence that increasing the content of irisin in the brain is important or is effective in replenishing, rescuing memory in mouse models of Alzheimer's disease. So we used 3 different mouse models of Alzheimer's disease, and we found basically the same result. I won't have time to show you everything here now. But of course, we submitted this paper, and then an outstanding question was, okay, exercise protects mice against Alzheimer's disease. Irisin protects mice against Alzheimer's disease, but does the effect of exercise depend on irisin? And that's why we did this experiment here that was requested by one of the reviewers. It's a complex experiment, but I'll try to keep it simple. We did basically the same memory test here that I showed you before, comparing the old and the new object. But we had here 3 conditions: mice that received A beta oligomers, mice that exercised, and some that remained sedentary, and mice that had received an injection to block irisin. So focusing here on these final columns here, final bars here, we have evidence that those mice who exercised had protection in terms of memory impairment. But those who exercised and received the injection of an antibody to block irisin were no longer benefited by this effect of exercise. So that's not the best experiment. If we could, we would have used knockout mice, but that's an important evidence. That's an important piece of evidence to show that irisin mediates the beneficial effects of exercise in the brains of mice that have AD symptoms, Alzheimer's disease symptoms. And of course, this story was published in Nature Medicine a couple of years ago. It received a lot of attention. It has received a lot of attention. I think it has many important aspects, because not only it stimulates policies to stimulate exercise in different populations, but it also offers a window or a target to stimulate neuroprotection in the patients, those patients who are already developing some symptoms of Alzheimer's disease. Okay, but how does irisin work in the brain? Is there any glimpse into signaling or mechanisms as to how irisin works? So we did some experiments, and what we found is that irisin quickly or rapidly stimulates ERK signaling, ERK phosphorylation. It is quantified here. And it also protects neurons against oxidative stress. So we use DCF as a probe for reactive oxygen species. And what we already knew, that A beta oligomers trigger in the accumulation of reactive oxygen species in neurons. And this is fully protected by irisin treatment. That was done by Guilherme, who was a student in the lab. And just to shift gears here, just to show that in another project that we started during that quarantine 3 years ago, when we were all suffering with COVID and could not be in the lab, we observed that in patients, there is a SNP, there is a single nucleotide polymorphism that causes the brain to slightly accumulate more A beta oligomers, A beta in the brain. So it may tell us that this slight accumulation of A beta in the brain, a slight increased accumulation of A beta in the brain, may predispose the patient for future accumulation. Of course, if you look at these 2 violin plots here, they look very, very similar. So the effect is really, really slight, small. But given the type of cohort that we're using here, it may tell us that combined with another factors, with other lifestyle factors, it may be another risk factor for future dementia. And we also found that these patients who harbored this SNP, they had glucose hypometabolism in the brain, especially in areas of the brain that are important for memory. So that was done in collaboration with Andrea Benedetti and Eduardo Zimmer. And that's an interesting result, because despite of massive beta amyloid accumulation, we can recapitulate features of Alzheimer's disease in patients who are not diagnosed, or especially diagnosed with Alzheimer's disease, but they carry this SNP single nucleotide polymorphism that may predispose patients for future dementia. Let's say that. Okay, so that's a small glimpse into how irisin works in the brain. And I have to acknowledge that irisin is not the only exercise-linked molecule, and it's not the only factor of exercise signaling, of exercise-mediated signaling. We published this review a year and a half ago, showing that there are many molecules that are induced by exercise systemically, and that they act in the brain to promote neuroprotection, synaptogenesis, and to promote memory and to control mood. So it's really important that we adopt practices of physical exercise as a populational approach to reduce or to avoid the burden of dementia in the future. And that's also important because this has been demonstrated, let's say, epidemiologically. So patients who have a positive lifestyle, a favorable lifestyle practices, even if they have higher genetic risk for Alzheimer's disease, they are protected. They have reduced risk of developing dementia. So that's really important. But you may ask me, so what is this favorable lifestyle that you're saying? Not smoking, of course, exercising, having a good nutrition, or basically have a favorable diet, not eating too much meat, not drinking too much, not eating too much ultra-processed food, and of course, engaging in activities that are good for the brain. Puzzles, or reading, playing music, playing some musical instrument, or something like that. So all of them combined comprise these favorable lifestyle factors that protect against dementia. And back again to the inflammatory story, I'll just talk about for more couple of minutes to show that I'm still, and I'm even more and more interested in inflammatory signaling in Alzheimer's disease. And that's something we're getting back to the bench to study a bit more. So I showed you this slide before showing that all these risk factors, all these genetic risk factors for Alzheimer's disease, they somehow relate to inflammation. And we became interested because of Fabrizio Pamplona, who is a researcher in the field of cannabinoid research, and who published this beautiful paper 10 years ago, showing that lipoxin A4, which is a resolving molecule, a lipid mediator that is known to resolve inflammation systemically, but lipoxin A4 appears to enhance cannabinoid receptor 1, CB1, in the brain. So this description by Fabrizio and Renato Takahashi was a landmark discovery because it showed a different function for lipoxin in the brain rather than only resolving inflammation. So we tried to, in a collaboration with Fabrizio and with Fernando Tovarmol and Claudio Canetti, we decided to study what happens to lipoxin A4 in dementia. So the first thing that we found was that the levels of lipoxin in the CSF, in the cerebrospinal fluid of dementia patients, was reduced, as you can see here, when we compare them to healthy controls and to people with early cognitive impairment, mild cognitive impairment. And the levels of lipoxin also correlate positively with memory. So the higher the levels of lipoxin A4, the higher the memory capacity here in humans. And it also correlates with BDNF, which is an important neurotrophin to the brain, and it also correlates with the beta-42 to a minor extent, but still correlates. So that tells us that lipoxin A4 may be also important to the brain, and that's why we decided to study that in mice. Mice that are knockout for 5-lipoxygenase, which is the rate-limiting enzyme for lipoxin production, lipoxin A4 production, they are better at, they are impaired in terms of learning, so they take longer to learn a task, and they also have reduced capacity of retaining the short-term memory. And they also have problems in forgetting, let's say, or in changing their memory. They are also impaired in terms of their memory extinction. So they take longer to form a new memory to overcome a previous one. So that's what we call extinction. So it looks like lipoxygenase controls somehow memory, and it may be related to lipoxygenase 4. Of course, lipoxygenase has many other products, not only lipoxygenase 4, but still it looks like promising data that we want to explore in the future. And this paper was published last year. And when we challenge mice with LPS to cause an inflammatory response, and of course trigger memory loss, we observed the expected phenotype of memory loss here. So mice in contextual fear conditioning, they have reduced freezing, which is the expected response. But when they are administered lipoxygenase 4, they are protected from memory impairment. And they are also protected from neuroinflammation. So the level of IL-1 beta increases with LPS in the brain, that's expected, but most mice are protected from this increase when they receive lipoxygenase 4. So that tells us that lipoxygenase 4 in the brain may rescue this inflammatory related memory phenotype. Okay, I won't tell you a lot about this quarantine story now because I don't have much time here, but I think I'll open a bit for discussion. And I'll conclude here by saying that inflammation is part of Alzheimer's disease response. We showed many years ago that is mediated by TNF-alpha, but of course there are other mediators that still need to be understood and studied. This exercise-related myokine called irisin is important for exercise neuroprotection in Alzheimer's disease. Lipoxygenase 4, this lipid-derived mediator of inflammation resolution, is reduced in the AD brains and CSF, and its loss is important to trigger memory impairment. And as a global conclusion here, it's important to study more and more the impact of hormones and metabolites in the context of cognition and memory, because we always thought of Alzheimer's disease as a brain disease, but of course we have now to consider that the system, the physiology of the whole body is important to control memory, and of course it may be impacted in the context of Alzheimer's disease. So peripheral energy metabolism, and of course peripheral hormonal signaling is important, is essential to control what's happening in the brain, and of course to mediate, to increase or decrease the risk of causing Alzheimer's disease. So of course, if we find effective therapies to treat dementia, we have to consider that these peripheral molecules are important regulators of brain function. So I have to acknowledge many of my students and collaborators. Without them, it would be impossible to carry out all this research. We have many collaborators here at UFRJ, and of course at other universities in Brazil and in the United States, and of course in Sweden now, where Andrea Benedetti is working. And I have to thank the funding agencies, FAPERJ, here from Rio de Janeiro, CNVQ nationally, and of course the private funding agencies, Alzheimer's Association, Serra Pilheira Institute, which have been essential for our research here, IBRO, and the International Society for Neurochemistry for funding our research. So I'm part of a big lab, which has 3 professors, and we work very closely. We work very in close collaboration to try to tackle these challenges in Alzheimer's disease. Of course, there are other lines that we're pursuing related to inflammation, to metabolism, to biomarkers, but I thought it would be more interesting to show you some metabolic and inflammatory stories today. And of course, if there are candidates for a PhD, for a master, for a postdoc, we are recruiting. So we travel a lot. We go to conferences, to international conferences. We are a very nice team. So if there's anyone interested in working with us here in Rio de Janeiro, which is a wonderful city, please email me or contact me on Twitter, or just drop me a line and I'll be happy to talk. So again, thank you very much for having me here. It was a pleasure to show you some results, and I'm of course open for discussion.
Thank you very much, Michelle. It's amazing work. Really nice to see it all put together like that, and also see that you have all those different lines that you carry on. So it's nice that the video is gonna stay on our channel on YouTube. So also you're opening for applications. So that's very interesting. And we have a tradition that we usually ask the students first if they have any questions. So we'll keep on that, and then we go on to the professors and postdocs. And so if there are any questions online as well, if you could raise your hand, I can see from here if there is anything. So sometimes we need a little bit of time to start with juicing up our brains, I guess. So I think I'm gonna just start with the question. One of the things is that you showed that there is some correlation, or at least you can see the development of Alzheimer's disease depending on some mutations, right? And you showed that graphically. And I wonder if you have looked at irisin production, if it correlates with those with if you're a carrier, if you have a mutation, if you have a difference in terms of expression or generation of the amyloids, if you also have lower irisin, or if there's something that you'd suggest as a supplement that you could type yourself, let's say, to try to avoid besides doing exercise, or if these people they tend to produce less irisin within doing the same exercise amount, like if those could be also correlated with disease development or some predisposition to developing disease.
That's a very good question, Lina. Well, we know that there's patients with symptoms of Alzheimer's disease, they have less irisin in their brains. But patients with mutations are a bit more difficult to find because they are rare, and there are many different mutations. So the genetic or the hereditary component of Alzheimer's disease is rare. So basically 1 to 2% of all Alzheimer's disease cases have a mutation causing it. So it's not easy to find them. And of course, there are some groups who investigate those cases. There's a large family in Colombia, for example, harboring a mutation for Alzheimer's disease, and there's a group studying them, but it's not easy to have biofluids or samples from them to measure irisin. So that's something I don't know. We know from our cohort that patients harboring the ApoE4 allele, they do not have different levels of irisin. So the association of irisin is higher with memory impairment rather than with the presence of this mutation of ApoE4. This variant, actually. So, but of course, given that the attention and the importance that irisin has gained over the past few years, I'm pretty sure that someone with access to a large cohort of mutant or to patients with familial Alzheimer's disease will study that soon. I bet probably we have the answer in a couple of years.
Thank you. Thank you. Gustavo, there was a person that I thought was a student, but I think lowered the hand. So Gustavo, do you want to ask from there, and we'll be alternating Rio and São Paulo?
Yeah, all right. Thank you so much, Michelle. Wonderful work. I'm curious about the role of cell death of these, central nervous system cells. So if you can comment a little bit about that, and in the terms also of the irisin anti-inflammatory, drugs, does it regulate cell death, or it helps like plasticity also of the neurons?
Yeah, that's a really good question, Gustavo, because when the first hypothesis of Alzheimer's disease came out, it was thought that cell death was the major driver of memory impairments. We now know that cell death is important, of course, especially when the disease is already full-blown or in place. But there's something going on before evidence cell death happens. So, I mean, there's synapse loss. Loss of synapses seems to be the best correlate to memory impairment patients. So the neuron is not dead, but it loses its synapses, its connection, it loses function, and it will eventually die, of course. So we have to keep this neuron alive because it will not be replaced, because neurons generally do not divide. So our goal here is to prevent the events that lead to these initial signaling mechanisms that cause eventually cell death. There are some papers showing that irisin prevents against cell death, neuronal death, but I think we have to act before that. Actually, at the moment that irisin can protect against toxic signaling rather than apoptosis, for example. I don't know if I answered your question, but.
Thank you, Michelle. Professor Julie Sarfestein would like to make a question.
Hi, Michelle. I was so glad because I had the privilege to see your presentation in our institute several years ago. It was actually the first time that I heard about irisin, and then we saw the paper. So it's great to see that you have your lab and the whole group is working so. It's a wonderful, wonderful project. Let me ask you some questions that I'm sure that you are discussing in the group. Because we are talking about systemic manifestations that probably precede the development of the severe manifestations of Alzheimer. And in the phenotypes that you have described with the lipoxygenase, they lead us to think about what happens with the endothelial cells. And I don't know anything about the regulation of irisin production by the endothelial cells themselves. I don't know anything about what happens with the blood barrier, because you have models that are very interesting. You are working with cardiovascular exercises, so we don't know if there are discrepant regulation of the release of irisin by microcirculation in different organs. And maybe I'm missing some basic facts about that. I saw in your skin something about cathepsin B being involved into, I'm not sure whether you are talking about the release of irisin by itself. So let's go to molecular, okay? My question is, what is known about the processing, the regulation of the processing events that lead to irisin liberation? And then maybe you can interconnect this with what is known about the blood barrier. So that's it. Vascular. Oh, this is.
Thank you very much for the question. I remember you were in the audience a few years ago when I talked at the Biophysics Institute. So thank you for being here again. Well, a couple of basic information about irisin. Irisin is mostly produced by the skeletal muscle and the brain. So the skeletal muscle is the major producer of irisin in the body, and the second place by a half levels of irisin is produced by the brain. Although it was described 10 years ago, now that we know a bit of basic irisin biology, let's say that. So it is now thought to cross the blood-brain barrier. So it has some interaction with endothelial cells at some point. We had experiments showing that delivering irisin in the periphery, in the blood, made positive effects in the brain, but we didn't know if it was crossing the blood-brain barrier or if it was having indirect effects. But now we know it crosses the blood-brain barrier. We don't know exactly the protease that cleaves FNC5 into irisin. There's some argument there's some debate as to whether ADAM10 is the protease or even cathepsin. Cathepsin B itself was shown to be a myokine. It circulates although it's previously known as a protease, endosomal molecule. It is known to be found in circulation and also to have positive effects by exercise. So it's a confusing field. We still haven't got the answer clear, but we know that irisin by muscle, it is clear, gets into circulation, is increased by physical exercise, and also that ADAM10 can be potentially protease into irisin secretion. So that's what we know about. I don't know if I addressed your question.
Thank you, Michelle. So I'll just I'll ask Claudia to hold just a little bit because Lucas Bolini is one of our students in the program. So I'll just jump him ahead on that theme of having the students asking before the professors, and then we'll go back to Claudia. Lucas.
Thank you for this amazing work. I was wondering, we know that astrocytes can acquire a very inflammatory phenotype, the A1 astrocytes that Ben Barry's described in the past 10 years. And it's also known that astrocytes in general are able to produce lipoxins. So I was wondering if you know whether irisin would interfere with the astrocyte phenotype that is found in Alzheimer's disease and other neurodegenerative diseases, if it could prevent the A1 astrocyte phenotype formation and then also help increase lipoxin production?
That's a very good question. There was a paper by the group of Christian Run from Harvard in 2021 showing that irisin may interact with astrocytes, yes. But it was not a thorough characterization of astrocyte response to, let's say, Alzheimer's and then even protective effects of irisin. That's something we are doing in the lab now. I still haven't got the answer, but it's most likely, yeah, I think so. And of course, there's this potential interaction between irisin controlling lipoxin production and that triggering positive effects in the brain. Yeah, that's a very attractive hypothesis, I think. Thank you, Lucas.
Thank you, Lucas. Thank you, Michelle. Claudia, thank you for waiting.
Let me see. Can you listen to me?
We can hear you, the video. Okay.
Oh, nice, Michelle. Great talk and great data. Thank you very much. My question is more about detection. I think me and all the world we are anxious about early detection of Alzheimer's disease. I read recently about the tau protein. So the tau protein that comes from the brain. Do you think it's feasible we are going to have this in Brazil, or you are looking for it like to detect the concentration of tau protein, and can be like correlated like that? If it's high, great chance to have Alzheimer's.
I think that the field of Alzheimer's disease has been really exciting over these past few years because we've had many developments that it looked like in the past that like we were stuck, and now we moved forward, I think. So first, I did not mention it, but in January there was the first approval of a drug called lecanemab, which is the first drug to show some positive effects in patients. So it's not a cure again, but it reduced by 27% the rate of cognitive decline in patients. So that's exciting because it targets beta amyloid and it shows that we are in the right direction, I think. Oh. Second, biomarker. Over the past 5 years, many tests have been developed to detect tau in the blood. So the residues are debatable. Let's say we can discuss which phosphorylated residue is better, But in combination, looks like we are moving to a direction in which blood tests will be available in the next few years. Of course, there are some challenges for Brazil because we have an admixed population. Our population is ethnically different from those who developed the tests in Sweden or in Canada or in the United States. We have to know if this test will work as expected for our population. So that's important. And the second point is that we have challenges to implement this technology because it depends on single molecule analysis, SIMOA. So it's too expensive for us. But of course, I'm pretty sure that if the tests are approved for human use, there will be some efforts to try to accelerate and make it cheaper for testing, because no one will want to pay $10,000. You have $10,000 to pay for a blood test that may show that you do not have Alzheimer's disease. But I see a brighter future in which we have tests that early detect Alzheimer's. But of course, we have early detection, but it has to be coupled to an early treatment or an early intervention for those who are at risk.
Exactly, yeah. But it's not worth only to know that you are going to have, but what can we do for, yes, what can we do to prevent or to delay a little bit the Alzheimer's? So, but I would be one that I would try to see if I have tau proteins. Okay. Oh, thank you very much.
Thank you, Claudia. Thank you, Michelle. And on that note, I wonder if there's a lot of talk, and actually there's some, I think, personal experience of how people after the pandemic and in some ways associated with having tested positive for COVID have been having problems with memory, right? And I wonder, and now thinking about that, like muscle and the brain are the ones who produce irisin, and also the people who eventually got isolated also had a decrease in muscle mass, right? Because of lack of movement and all. And I know that is a very tough because of the multi-parametric nature of the problem. But I wonder, I just wanted to see your opinion about those things, about if you do see more, I really don't know, more disease in, let's say, women that tend to have less muscle mass, or if you have less irisin in people who have less amount of muscle than people who are more worked out, let's say, or if you see any correlation on the direction of the topics that you are studying that could explain or bring some light into those problems.
Well, I think that reduced irisin comes from two factors. Of course, sarcopenia, so reduced muscle mass content. It's frequent with aging, of course. But the second point, there is reduced signaling that leads to, even if we consider the total or the reduction in muscle mass, there will be net a reduction in irisin production. So I think there's two explanations for that. And of course, sarcopenia is relatively common in elderly people, but not all elderly people develop Alzheimer's disease. So there's some difference there. But of course, working out to prevent muscle mass loss is important to prevent cognitive decline. Even if this cognitive decline is not related to Alzheimer's disease, but only the age-related cognitive decline, which often happens. And patients with severe sarcopenia, with autoimmune disorders or with mutations that lead to muscle loss, they have cognitive impairments. So I do believe that there is a connection between the content of muscle mass and muscle physiology and cognitive function. But I don't think it's only because of irisin. There are other factors secreted by the muscle, IL-6, for example, and others that are important for the brain too.
It's just a curiosity because I think that it was early this year, maybe by Christmas time, when the market was very disappointed because they were testing a humanized antibody. I think it was against the oligomer, right? And there was this famous case of a patient that really, I mean, died, but it was fulminating attack, and there was so much publicity about that. And I just wonder what people think that has happened with this patient. Was it, a coincidence? Because there was a major expectation that, this could be a step forward. And I don't know what you and Sergio and Fernanda, what are your thoughts about that?
Well, actually, to place a historic context, there have been many clinical trials with antibodies targeting A-beta, many forms of A-beta, and all of them failed, basically. Most of them failed, either because they caused some adverse effects, so the trial had to be stopped or halted, or because they didn't work. The drug didn't work, the antibody didn't work. We believe that that's caused by the lack of correct specificity of the antibodies. So if you produce an antibody against A-beta, it will mostly bind to plaques, to amyloid plaques, and they will be stuck on that plaque, on those plaques, and they probably will not be able to diffuse and target the soluble species, for example. So there's been more and more optimization to develop antibodies that bind more soluble species, because those are the ones will bind the synapses and interfere with synapse communication. So those are the species that we want to target. And this recently approved drug, the lecanemab, it also binds plaques, but it binds soluble A-beta as well. So the pharmacy field is trying to optimize those antibodies so that they could bind more soluble species. And we believe that that will be more effective. There was the aducanumab, the previously approved drug, which was approved among some controversies because of some business issues that looked like the business was more important than the science, because the FDA panel recommended against approval, but even so the director of FDA and the board, the executive board approved the drug anyway. And it had no clear statistical effect, and it also caused some adverse effects in some patients, and that was a real matter of concern. In addition to the fact that the drug is really expensive, it costs $56,000 a year. So it's complicated. But the point is, targeting beta amyloid excessively is a problem, because it may bring adverse effects, brain edemas and other memory effects, can even worse the disease. But if you have the right amount of specificity and the right amount of efficacy, I think it's the right target to follow. That's what I would do. And that's what lecanemab is showing now.
Okay. As an immunologist, I'm just curious if you could tell us whether all the humanized antibodies like this one that cause these terrible effects, they were lacking FC, so you would not have, let's say, the potential to activate either the complement system or even interacting with FC receptors. So from the, looking to this problem from the basic standpoint, it's very weird, that you have a tremendous reaction in a question of minutes. That's what happened with the patients. So that's the reason I was so curious to know what you know about that. But thank you for the explanation.
Thank you.
And thank you very much, Michelle. I think if we do not have any other questions, let's see if somebody shows up in the web. But thank you so much for your time and for presenting to us. I know that there are a lot of professors that were really sad and said they're going to watch your talk on the YouTube later because it conflicts with the neurotox from ICB, the PAN. So they were asking, so I'm sure that you might get some questions afterwards and people will be assessing your talk. So thank you very much again. And it was really great to hear about your talk.
Yeah, I'm really happy to be here. Thank you for the invitation again. It's good to talk to a different audience. It's good to see that this multi or inter-institutional events with physics and WISP, so that's good to have this different experience. And it's good to see students here and students asking. So if your students out there, do not feel ashamed to ask. Go and ask questions. It's really important to do so. And thank you. Sorry, maybe I ran away of time, but I think we had a good conversation here. And if you have any questions, please email me and drop me a line. I'll be happy to answer. I'm kind of cold today, so it's not the best day, best presentation I had, but I hope you could get the message, and I hope you understand this piece, this brick we're trying to build this whole long. Thank you very much.
It was really great. If you hadn't mentioned anything, we wouldn't have noticed. You really gave a great talk, and it was a great discussion. Thank you very much.
Thank you.
Bye-bye, pessoal. See you later. And on next week, just for the students, and we won't be having the webinars because it's Holy Week. So USP stops for the week, so we'll be stopping together. So we resume on the 10th of April. Is that right? I think Louisa, but we'll as soon as we have the next ad, we will share. Okay, so thank you.